Hello students, welcome to EPG Pathshala. I am Dr. Divya Bajaj from Hindu College, University of Delhi. Today we are going to discuss about the module Clinical Immunoassays, including the techniques ELISA, RIA, FACS and Immunofluorescence from the paper Immunology. After studying this module, you shall be able to define the basic characteristics of an immunoassay, understand how a target molecule can be detected and quantitated, how different types of labels can be employed to detect immune reactions, how a wide array of information can be obtained relevant to the nature and localization of the target antigens, differentiate between direct and indirect detection methods, and basic principles and clinical applications of immunoassays. Immunoassays. An immunoassay is a biochemical test used to identify the presence or amount of a particular molecule referred as an analyte in a solution by combining with an antibody or an antigen. The principle of immunoassays is formation of an immune complex involving the recognition and binding of an antibody to a specific molecule among a mixture of molecules. A key feature for all immunoassays is generation of a measurable signal in response to the binding. Immunoassays utilize a wide range of labels, some emitting radiation, resulting in a visible color change, fluorescence under light, or could be induced to emit light. Examples of the applications of immunoassay include drug testing, hormone testing, for example, insulin in diabetic patients, bacterial or viral testing, for example, AIDS or hepatitis, environmental testing, for example, testing of herbicides and pesticides. Advantages of immunoassays include these are inexpensive, highly selective, they have low limits of detection, high throughput applications, applicable to the determination of a wide range of compounds. This slide demonstrates the basic principle behind an immunoassay. The detectable label can be attached to either of the components, the antibody or the antigen, participating in an immunoassay. We can see in the diagram that in the first half, the label is attached to the Fc arm of the antibody, which is reacting with the analyte in the specimen, and it leads to the formation of an antibody-antigen complex, which can be detected with the help of the label. In the second half, however, the label is attached to the antigen, which is reacting with the antibody, forming the antigen-antibody complex, and this can be detected with the help of the label. Categories of immunoassays. The first category, or the competitive assay or limited reagent assay, involves measurement of an unlabeled analyte or antigen by its ability to compete with the labeled antigen in the immunoassay. Higher the reduction in the label at the end of the assay, greater is the amount of antigen in the test sample. The second category, or the non-competitive immunoassay, can be divided into one-site non-competitive, where the unknown analyte in the sample are allowed to react with labeled antibodies. After binding reaction is complete, the unbound antibodies are washed away and the bound label antibodies are measured as signals for the complexes formed. Intensity of the signal is directly proportional to the concentration of unknown antigen. The second, or the two-site non-competitive immunoassay, involves absorption of an antibody on the solid phase surface when it is allowed to interact with the test sample containing the antigen. The labeled antibodies are in excess in this system and bind specifically to their respective analyte. Subsequently, a second label antibody is added, causing sandwiching of the target. This slide shows the different types of labels which can be employed in immunoassays. A label is a molecule that reacts as a part of the assay and in doing so produces a signal that can be measured in the solution. Different types of labels are available for immunoassays, like radio labels where radioactivity is measured, fluorescent labels where fluorescence is measured, enzymatic labels which result in formation of colored products with the help of enzyme-substrate reactions, and chemiluminescent labels which result in light production with the help of a chemical reaction. This slide shows the pros and cons of direct versus indirect labeling. First, we look at direct labeling. It is a faster process because it uses only one antibody and avoids non-specific binding of secondary antibody. But immunoreactivity of the primary antibody could be compromised as a result of labeling, and the signal could also be less intense. When we look at indirect labeling, enhanced sensitivity is achieved as a result when primary antibody contains several epitopes that combine with the labeled secondary antibody, resulting in signal amplification. However, non-specific binding may occur during the addition of secondary antibody. Extra incubation and wash steps also make the process time-consuming. This slide shows the technique ELISA, or Enzyme-Linked Immunosorbent Assay. It was first developed by Aramis and Pearce. It involves an antibody-coupled enzyme which reacts with a colorless substrate called a chromogenic substrate, resulting in the generation of a visible colored reaction product. The enzymes commonly employed for ELISA include alkaline phosphatase or AP, horseradish peroxidase or HRP, and galactosidase. These assays possess high sensitivity and are safe and cost effective. The result generated from an ELISA assay could be qualitative, identifying the presence or absence of a particular antigen molecule. It could be semi-quantitative, analyzing relative antigen amounts in assay samples, or quantitative, defining precise antigen concentrations with respect to a standard curve. There are different variants of ELISA known as indirect ELISA, sandwich ELISA, and competitive ELISA. Indirect ELISA. Indirect ELISA is used for both qualitative and quantitative measurement of antibodies. It involves addition of the sample solution containing a primary antibody to a microtiter plate well coated with the antigen, such that the antibody would react with this well-bound antigen. The unbound antibody is washed away, and this is followed by detection of the antibody bound to the antigen with the help of an enzyme conjugated secondary anti-isotype antibody. This specifically binds to the primary antibody. The unbound secondary antibody is also washed away, followed by addition of substrate for the enzyme. The colored reaction product is analyzed spectrophotometrically by plate readers. This slide shows the technique indirect ELISA. Antibody can be detected or quantitatively determined by using this technique. Here, antigen is coated on the microtiter well as shown in the figure. Serum or some other sample containing the primary antibody is added to the microtiter well and allowed to react with the coated antigen. Any free primary antibody is washed away and the bound antibody to the antigen is detected by adding an enzyme-coated secondary antibody that binds to the primary antibody. The unbound secondary antibody is then washed away and a specific substrate for the enzyme is added. The enzyme hydrolyzes the substrate to form colored products. The amount of the colored end product is analyzed spectrophotometrically by measuring the absorbance of all the wells of the microtiter plate. The rate of color formation is directly proportional to the amount of the specific antibody. Sandwich ELISA. Sandwich ELISA is used for quantitative or qualitative analysis of antigens. The basic principle of this technique is the antigen-antibody interaction. But here the antibody instead of the antigen is immobilized on the microtiter well. An antigen sample is then added to the well pre-coated with the immobilized antibody. The excess or unbound antigen is washed off using buffers, followed by addition of a second enzyme-linked antibody which is specific to a second epitope on the bound antigen. Unbound secondary antibody is then washed off and a substrate corresponding to the enzyme on the secondary antibody is added. And the colored reaction product can be analyzed spectrophotometrically. This slide shows the pictorial depiction for the technique Sandwich ELISA. Here we can see that the antibody instead of the antigen is immobilized on the microtiter well. The antigen sample or protein is then added to the well pre-coated with the immobilized antibody. The excess or unbound antigen is washed off using buffers. This is followed by the addition of a second enzyme-linked antibody specific to a second epitope on the bound antigen. The unbound secondary antibody is then washed off. A substrate corresponding to the enzyme on the secondary antibody is added next. The colored reaction product formed as a result of the enzyme substrate complex is measured spectrophotometrically. This slide shows the pictorial depiction of the technique competitive ELISA. The first figure shows that the antibody is pre-incubated in solution with a sample containing the antigen. This antigen-antibody mixture is then added to the microtiter well pre-coated with the same antigen, shown in the next. The more the antigen present in the sample, lesser is the free antibody available to bind to the antigen-coated well, hence the name competitive. After the well is washed with a buffer, enzyme-conjugated secondary antibody specific for the isotype of the primary antibody is added to determine the amount of primary antibody bound to the well. After washing the secondary antibody which is unbound, the substrate is added which is specific to the secondary antibody. This slide shows the comparison between different variants of ELISA. ELISA can be performed with a number of modifications to the basic procedure, and the different procedures are shown here, including direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. The key step or the immobilization of the antigen of interest can be achieved by direct absorption to the assay plate or indirectly via a capture antibody which has been attached to the plate. The antigen is then detected either directly using an enzyme-labeled primary antibody or indirectly using an enzyme-labeled secondary antibody. Irrespective of the means by which the antigen is captured to the plate, the sensitivity of an ELISA assay is largely determined by the detection step. The detection antibodies are usually labeled with alkaline phosphatase or horseradish peroxidase. A large variety of substrates are available for performing ELISA with the help of an HRP or AP conjugated antibody. This slide demonstrates the technique chemiluminescence. Chemiluminescence is an ELISA variant where an enzyme-substrate reaction produces a byproduct that emits light instead of developing a visible color. Luminescence is defined as emission of light from a body as it returns from an electronically excited state to ground state. Chemiluminescence consists of measurement of light during a chemical reaction. Like we can see on the figure on the left side, oxidation of the compound luminol by hydrogen peroxide occurs and the enzyme HRP produces light. When the luminol substrate is added to the microtiter plate well, which contains the capture antibody bound to the HRP coated antibody, the resultant reaction emits light. Advantage of this assay is enhanced sensitivity. The detection limit of a luxogenic substrate or a light-emitting substrate is at least 10-fold higher than a chromogenic one. It could be increased to more than 200 folds using enhancing agents. The Elispot or the Enzyme-Linked Immunospot Assay. It was developed by Cecil Czerny's group in Gothenburg, Sweden, 1983, in order to detect antigen-specific antibody-producing cells. It helps in identification and quantification of the number of cells in a population producing antibodies in response to a particular antigen or vice versa. Adsorption of the plates with antigen called the capture antigen is followed by reaction with the antibody of interest or with the antibody specific to the target antigen. A population of cells to be analyzed is then added in the form of a suspension onto the treated plates and incubated. The cells collect at the surface of plates and bind to the secreted substances specific to these capture molecules, thus producing a ring of antigen-antibody complexes around the cell secreting the molecule of interest. The unbound molecules are washed off and an enzyme-linked antibody specific for the secreted antigen is allowed to bind. The assay is subsequently developed by chromogenic or chemiluminescent substrates, identifying each antibody or antigen-producing cell indicated by color or light. A substrate is selected such that a precipitate is produced instead of a soluble product, displaying the result in the form of visible spots corresponding to each antigen secreting cell. This slide depicts the procedure for the technique Elispot assay. Elispot assay is used for identification and quantitation of the number of cells in a population which produce antibodies in response to a particular antigen or vice versa. The procedure of this assay involves first, incubation of antigen-secreting cells in wells which are pre-coated with specific antibodies. Next, the cells are removed by washing and the secreted analyte or antigen is captured by the immobilized antibody. After this, it is incubated with the biotinylated antibody. In the next step, Incubation is performed with alkaline phosphatase conjugated streptavidin antibody. Substrate is added next and the reaction product is obtained in the form of colored spots. The substrate is selected such that a precipitate is produced instead of a soluble product, displaying the result in the form of visible spots corresponding to each antigen secreting cell. This slide shows the technique radioimmunoassay or RIA. One of the most sensitive techniques for detecting antigen or antibody was first reported by S. A. Burson and Rosalind Yallow in 1960 in order to analyze the levels of insulin and anti-insulin complexes in diabetics. The technique demonstrates high sensitivity and is capable of quantitating hormones, serum proteins, drugs, and vitamins at concentrations as low as 0.001 micrograms per milliliter. The basic principle of this technique is competitive binding between the radiolabeled and unlabeled antigen to a high affinity antibody. First, the antibody is allowed to interact with the radiolabeled antigen, saturating the antigen binding sites of the antibody. This is followed by addition of large amounts of sample containing unknown amount of unlabeled antigen. The free binding sites on the antibody are available to both the labeled and unlabeled antigens as the antibody is unable to distinguish between the two. The labeled antigen is progressively displaced from the antibody binding sites with increasing amount of the unlabeled antigen. This reduction in the amount of radiolabeled antigen bound to the specific antibody on increasing antigen concentration in the unknown sample is measured in order to quantitate antigen concentrations in the test sample. This slide shows the pictorial representations for the technique radioimmunoassay. The basic principle of this technique is competitive binding between the radiolabeled and unlabeled antigen to a high affinity antibody. The technique involves steps where the first step is interaction of the antibody with the radiolabeled antigen such that the antigen binding sites of the antibody are saturated. This is followed by addition of large amounts of sample containing unknown amount of unlabeled antigen. The free binding sites on the antibody are available to both the labeled, shown in red, and unlabeled antigens, shown in blue, as the antibody is unable to distinguish between the two. The labeled antigen is progressively displaced from the antibody binding sites with increasing amount of unlabeled antigen. This reduction in the amount of radiolabeled antigen bound to specific antibody on increasing antigen concentration in the unknown sample is measured in order to quantitate antigen concentration in the test sample. Immunofluorescence. Albert Koons first demonstrated the labeling of antibodies with fluorescent molecules in 1944. These molecules possess the inherent property of absorbing light of a particular wavelength and emitting light of another wavelength. Antibodies tagged with the fluorescent dye or fluorochrome can be identified by emission of colored light when excited by light of a specific wavelength when these are a part of immune complexes. Molecules with luminescent properties emit light of a different wavelength on absorbing light of a particular wavelength. Fluorescent materials give off light very promptly due to their atomic structure. The light emitted from luminescent objects can be visualized using a fluorescence microscope equipped with a UV light source. These fluorescent probes are conjugated to the Fc arm of the antibody molecule, ensuring that the specificity is not affected. An important application of this technique is detection of antibodies bound to antigen epitopes in cell cultures or tissue sections. This slide shows the pictorial depiction of the technique immunofluorescence. The left side of the technique is showing primary immunofluorescence, where a primary antibody is conjugated with a fluorophore and it is reacting with the antigen, producing the antigen antibody complex, which can be detected with the help of measuring the fluorescence produced by the label. In the second half, a similar demonstration is given for secondary immunofluorescence. For secondary immunofluorescence, the antigen is bound by the primary antibody, which is in turn bound with the secondary antibody conjugated with a fluorescent label probe. The antigen antibody complex can be detected again by measuring the fluorescence produced as a result of the fluorescent label. Flow cytometry. Flow cytometry aims at automation of the process of cell type segregation of immunostained cells and their subsequent analysis. A flow cytometer utilizes a laser beam and a light detector for measuring the cell signals in suspension, hence allowing quantitation studies. Each time the laser beam passes through the cell, the light coming from the deflector is deflected from its path, and this intermittent disconnect of the laser signal is then recorded. The cells in suspension are tagged with the fluorescent label by direct or indirect immunofluorescence methods. This is followed by analysis of these cells on the flow cytometer. Cells bound to fluorescently labeled antibodies get excited on being hit with the laser beam and emit light, which is subsequently recorded by a detector unit installed at the right angle to the laser beam. These signals are detected towards a computer which generates plots of cell number versus their fluorescence intensities. Advanced instruments are also involved in the cell sorting into different locations according to their fluorescence intensity profiles. The output investigates the number and type of every cell in a population that binds fluorescently tagged antibodies. This slide demonstrates FACS or Fluorescence-Activated Cell Sorter. Fluorescence-activated cell sorting is a technique based on the principle of flow cytometry, where cell sorting is performed on the basis of their protein constitution. It finds excessive application in immunology, where blood cell counts are measured in patients with HIV and leukemia, for diagnosis of the disease as well as monitoring the patient's response to treatment. Cell sorting is also effective in understanding the functions of drugs, development of drug resistance, detection and identification of viral antigens, and development of new drugs for various disease conditions. The FACS method can detect cells with as few as 3000 molecules of fluorescent dye bound to each cell. The cell size is also measured by means of correlated light scattering, such that the cells falling within particular ranges of both size and fluorescence can be separated. All cells in a multicellular organism contain identical DNA, but tremendous variations are present at the protein level. Therefore, this principle is utilized to separate cells that are phenotypically distinct. FACS can also be used to quantitate the number of cells expressing the proteins of interest and also the amount of protein expressed. This slide shows the pictorial depiction of the technique FACS. The principle of FACS for analysis and sorting of cell population using fluorescence at two wavelengths is demonstrated. We can clearly see in the figure that a mixture of cells in a liquid are exposed to a beam of laser light. The setup is such that each cell enters a single droplet as it leaves the nozzle tip. This drop is given an electronic charge depending upon the fluorescence of the cell inside the drop. The deflection plates then attract or repel the cells accordingly into different collection tubes. The collection tubes are collecting positive, negative, and uncharged cells in different tubes, also shown with different colors. The sorted cell populations are then analyzed to ensure successful cell sorting. These sorted cells can be cultured if they remain viable and uncontaminated. So students, let us summarize what we have learned in this module. The key features of an immunoassay include formation of an immune complex and production of a measurable signal in response to the binding, which can be detected and analyzed. Immunoassays employ a wide range of labels, some which emit radiation, which produce a visible color change or fluorescence under light, and these can be induced to emit light. A competitive assay or a limited reagent assay is one where the amount of unlabeled analyte or antigen is measured by its ability to compete with the labeled antigen in the immunoassay. The target cell type can be identified using a fluorescent antibody specific for a cell surface antigen, and the percentage of this cell type in the total cell population of interest can be identified. Luminescence is defined as the emission of light from a body as it returns from an electronically excited state to ground state. All cells in a multicellular organism contain identical DNA, but tremendous variations are present at the protein level. Therefore, this principle can be utilized to separate cells that are phenotypically distinct by the FACS technique. Thank you.